Hello, I am Professor Sims, and in this video I will discuss the urinary system. This is the 8th in a series of 10 lessons held as part of my Anatomy and Physiology 2 online laboratory course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. By the end of this lesson, you'll be able to identify the structures of the urinary system, understand the basic function of the urinary system structures, illustrate the macroscopic and microscopic structures of the kidneys, outline how blood is filtered in the kidney nephron, and understand how urinalysis can help diagnose diseases. The urinary system plays a vital role in keeping our internal environment stable. One of its primary functions is filtering the blood to remove waste products such as urea and creatinine, while maintaining the body's fluid balance, electrolyte levels, and acid-base equilibrium. The system is composed of the kidneys, ureters, urinary bladder, and urethra. The kidneys form urine by filtering and adjusting the composition of the blood. The ureters transport urine from the kidneys to the bladder, where it's stored until it is expelled through the urethra. Together, these structures ensure that waste is efficiently removed and that essential substances are conserved. In addition to waste removal, the urinary system contributes to other important physiological processes through hormonal mechanisms such as the release of renin and erythropoietin. It helps to regulate blood pressure and stimulates the production of red blood cells in the bone marrow. This means that the urinary system is not only a filter and a drainage network, but is also a key player in maintaining homeostasis and overall health. The kidneys are the primary organs of the urinary system, and their main job is to filter the blood, remove waste products, and also regulate water and electrolyte balances. They maintain the body's internal environment by deciding what gets excreted and what gets reabsorbed. Each kidney contains about 1 million microscopic filtering units called nephrons. Nephrons are the functional units responsible for forming urine through processes like filtration, reabsorption, and secretion. Structurally, the kidney has distinct regions, and you can see here in the diagram. The outer cortex contains the bulk of the nephrons, and then beneath it lies the medulla. This is organized into triangular renal pyramids, and these pyramids drain into small chambers called calyces, which then merge into the renal pelvis. The renal pelvis is a funnel-shaped structure that collects urine before it enters the ureter, and together these structures, they work in sequence to ensure that the wastes are eliminated and that the body's water and salt balance is maintained. Let's look more in detail at the nephron. The nephron is the kidney's microscopic powerhouse. This is the structure where urine is formed and blood composition is fine-tuned. Each nephron begins in the renal cortex with the renal corpuscle, which contains the glomerulus, a tuft of capillaries where filtration occurs. Blood arrives here through the afferent arteriole and exits through the efferent arteriole, setting up the next stage of processing. From the glomerulus, the filtered fluid enters the proximal convoluted tubule, where much of the water, ions, and nutrients are reabsorbed back into the bloodstream. The filtrate then flows into the loop of Henle, which dips into the medulla and helps create the concentration gradient that allows the kidney to conserve water. Next, the fluid enters the distal convoluted tubule, where further fine adjustments to electrolyte and pH balance occur, often under hormonal control. And then the filtrate drains into the collecting duct, which delivers urine to the renal pelvis. And surrounding all of this is an intricate network of vessels, peritubular capillaries, and in the medulla, the vas recta, which play a critical role in reabsorbing needed substances and maintaining the medullary gradient. Together, these tubular and vascular components allow the nephron to act as both a filter and somewhat of a recycler, again, ensuring homeostasis is maintained. So let's look a little bit at the blood flow through the nephron a little bit more in detail. So the blood is carefully organized to support filtration, reabsorption, and secretion, and it begins as the blood enters the nephron through the afferent arteriole, which delivers it to the glomerulus. Again, the glomerulus is a dense network of capillaries, and they are specialized for filtration. Here, the plasma and dissolved substances pass through the filtration membrane into the nephron's tubule, while larger proteins and blood cells remain in the circulation. Then the filtered blood leaves the glomerulus via the efferent arteriole, which is narrower than the afferent arteriole, and this difference in the diameter helps to maintain the high pressure needed for filtration. From the efferent arteriole, blood moves into one of two pathways. In the cortical nephrons, it branches into the peritubular capillaries, which surround the proximal and distal tubules and handle most reabsorption and secretion. In juxtamedullary nephrons, it forms the vasa recta, which are long, straight vessels that run alongside the loop of Henle and help preserve the kidney's concentration gradient. This vascular network ensures that the filtrated substances that need to be reclaimed, stuff like water, electrolytes, nutrients, are efficiently reabsorbed, while waste and excess ions can be secreted back into the tubule for excretion. So let's look just a little closer, as the start of the nephron's filtration process lies at the glomerulus, a compact tuft of capillaries nestled inside of the renal corpuscle. We saw that here. Blood arrives here through the afferent arteriole, and the high-pressure environment forces plasma and dissolved substances through the filtration membrane into the surrounding Bowman's capsule, which is this entire structure here. Is Bowman's capsule. Large proteins and blood cells are retained in the circulation, while smaller molecules enter the tubular system, as we saw in the previous slide. Blood exits the glomerulus via the efferent arteriole. So this is the afferent arteriole, and this is the efferent arteriole. And remember, the efferent arteriole is narrower than the afferent arteriole. This is a design that maintains filtration pressure, right? And then from there, the blood enters either the peritubular capillaries for cortical nephrons or the vasa recta for juxtamedullary nephrons. So this is a juxtamedullary nephron, and this is the juxtaglomerular cells here. And this allows for reabsorption and secretion to occur downstream. Wrapped around part of the glomerulus is the juxtaglomerular apparatus. This is a specialized structure where the distal convoluted tubule passes close to the afferent arteriole. It includes the macula densa, which senses sodium chloride levels in the filtrate, and the juxtaglomerular cells, which release renin in response to low blood pressure or low sodium. This hormonal control links kidney function directly to blood pressure regulation. So together, the glomerulus and the juxtaglomerular apparatus act as a gateway for filtration and also a key regulatory checkpoint for homeostasis. If we zoom in onto the filtration surfaces of the glomerulus, we will find the podocytes. Podocytes are specialized epithelial cells that wrap around the glomerular capillaries. Each podocyte extends numerous foot-like projections called pedicels that interlace to form narrow gaps known as filtration slits. So those are these here, these filtration slits. These slits are not open holes. They are bridged by slim diaphragms made of specialized proteins that act as a selective barrier. And together with the fenestrated capillary endothelium and the basement membrane, they create this filtration membrane. And that's what this looks like here. The basement membrane and these little fenestrations. The fenestrations in the capillary walls are tiny pores that allow water and small solutes to pass freely while keeping blood cells and most plasma proteins inside the circulation. So this design maximizes filtration efficiency and also protects the body from losing valuable proteins. In essence, the podocytes and fenestrations work together as a team. The fenestrations provide the first level of size-selective filtration, and then the basement membrane adds a charge-selective barrier. And then the podocytes and the little slits ensure that only substances small enough and appropriate for the filtrate make it into the nephron. Let's move on to the ureters. The ureters are paired muscular tubes responsible for carrying urine from the kidneys to the urinary bladder. So it's these structures here. There's the kidneys and bladder, and then down here we'll talk about the urethra in a bit. Each ureter begins at the renal pelvis, where the urine collected from the calyces funnels into the tube. Their walls contain smooth muscle that produces rhythmic wave-like contractions called peristalsis. These contractions move urine downward toward the bladder, and they work independently of gravity, meaning the urine transport is just as effective whether a person is standing, lying down, or even if they're standing upside down on their head. The ureters enter the bladder at an oblique angle, and this creates a natural one-way valve. As the bladder fills, pressure at these entry points helps to keep urine from flowing backward towards the kidneys, and it also protects against infection and damage. This combination of muscular action and then structural design ensures that urine is delivered efficiently and safely to the bladder for storage. The bladder is a hollow muscular organ designed to store urine until it's ready to be released. Located in the pelvic cavity, it can expand significantly as it fills and then return to its resting size after emptying. The wall of the bladder contains the detrusor muscle, which is this thick layer of smooth muscle fibers, and it's oriented in multiple directions. During urination, the detrusor muscle contracts in a coordinated way, increasing pressure inside the bladder and expelling urine through the urethra. The bladder's inner lining is made of transitional epithelium. Transitional epithelium is a specialized tissue visible in histology slides. You can see it here. This epithelium has the unique ability to stretch and change shape, and this allows the bladder to hold varying volumes of urine without tearing or leaking. So it's these muscular walls and the elastic lining make the bladder both a strong storage reservoir and a dynamic part of the urinary system capable of adjusting to the body's needs for fluid balance and waste removal. Here we have some figures that are showing where the bladder is located in females and in males, and then of course the surrounding structures. You can see here that the bladder is connected to the urethra, and that is where urine is expelled. In females, it's a rather short, narrow passageway, whereas in male, it's also quite narrow, but it's longer because the urethra goes from the bladder through the penis in males. So this short length of urethra here that you see in females, this being so short is why it's quite easy. If you see, like right here is the labia. It's quite easy for women to get yeast infections because there's very small distance here from if you're using the restroom and you happen to wipe from back to front, then you can easily be wiping bacteria right into this urethra and causing urinary tract infections or yeast infections. Let's talk a little bit about the flow of urine. Urine is formed in the glomerulus, which we saw. This is where the blood is filtered. The filtrate collects in the Bowman's capsule. From there, the fluid enters the proximal convoluted tubule, where most of the water, ions, and nutrients are reabsorbed back into the blood. Next, it moves into the loop of Henle, descending into the medulla to concentrate the filtrate. Then it ascends back towards the cortex to adjust the salt balance. The filtrate then passes through the distal convoluted tubule, where the final fine-tuning of electrolyte and pH balance occurs, often under hormonal influence. From here, the fluid, which is now considered urine, enters the collecting duct, which carries it through the medulla to the renal papilla. Urine then flows into the minor calyces, merges into the major calyces, and enters the renal pelvis. The ureter transports the urine to the urinary bladder for storage. Finally, when urination occurs, the bladder empties through the urethra, releasing the urine from the body. So this pathway ensures that waste and excess substances are efficiently removed while the body retains what it needs for balance and health. And you should know this pathway. So as usual for the pre-lab, begin by carefully reading. It's chapter 25 in the textbook. That's the structure and function of the urinary system. Focus on understanding how the kidneys, ureters, bladder, and urethra work together to filter blood, form urine, and maintain fluid and electrolyte balance. Then you will want to review the introduction and background sections of Chapter 8 in your lab manual. These sections provide context for the lab exercises. They will help you see the key structures and outline the objectives that you'll need to achieve. And then finally complete the pre-lab exercises 8.1 through 8.3. These exercises are designed to reinforce your understanding of the material and, as usual, prepare you for hands-on activities. In lab exercise 8.1, you'll explore the major structures of the urinary system using anatomical models. Begin by locating the kidneys and identifying the renal artery and renal vein, which supply blood to and from each kidney. You're going to want to observe the internal regions of the kidney, including the cortex and medulla, which house the nephrons and collecting structures. Next, you'll want to trace the path of urine from the kidney through the ureters to the urinary bladder and then through the urethra. And you want to note all of the structures, the linings of those structures. Pay attention to the trigone, which is the triangular area on the bladder floor that marks the opening of the ureters and urethra. You're also going to want to locate surrounding structures that support the urinary system, including the aorta, which delivers blood to the kidneys, the adrenal glands, which sit atop the kidneys. You will examine the layers of the urinary tract, including the adventitia on the outside of the epithelium lining the lumen. And then you should be able to confidently identify all of these structures in a model and understand their spatial relationships. So let's have a look at some example models. This slide here highlights the key anatomical structures of the urinary system using detailed models. First, you will focus on the kidney, the organ responsible for filtering blood and forming urine. Within the kidney, you will be able to see the nephrons. Nephrons, again, are functional units where filtration, reabsorption, and secretion occur. If you zoom in even further on the nephron, you will be able to see the Bowman's capsule. This surrounds the glomerulus and collects the filtrate produced during blood filtration. The Juxtaglomerular apparatus, shown here, is positioned near the glomerulus, where the distal convoluted tubule passes close to the afferent arteriole. This structure plays an important role in regulating blood pressure, controlling the rate of filtration, and hormone release. So by studying these models, you can observe how the nephron's microstructures integrate within the kidney's larger anatomy to maintain fluid and electrolyte balance, remove waste, regulate blood pressure, all that good stuff. But it is important to understand these spatial relationships. So you're going from large, medium to small. So this is within this, is within this, okay? So understand those spatial relationships. This slide here is a closer view of some of the urinary system structures. First, you again observe the kidney. Kidney is here within the abdomen. Notice how the renal cortex forms the outer layer, right? While the renal medulla contains the renal pyramids, which are separated by renal columns. And at the tip of each pyramid is the renal papilla. So these are the renal columns, cortex. You have a renal pyramid here. And then at the tip of these renal pyramids are the papilla. And of course, this is where the urine drains into a minor calyx, which then funnels into the renal pelvis. And from the renal pelvis, urine enters the ureter, which carries it to the urinary bladder for storage. By examining, this is the abdominal model, this model, and the isolated kidney model, you can see how these structures are organized spatially again, and how urine flows from formation in the kidney to transport through the ureter and eventual storage in the bladder. So knowing these spatial relationships is important for clarifying both normal urinary function and also the basis for potential clinical issues such as obstruction or reflux. In lab exercise 8.2, you'll explore the microscopic anatomy of the urinary system using figures 8.4 through 8.7 in your lab manual. Begin by observing the cortex and medulla of the kidney, which house the functional units of the nephron, and pay attention to the loop of Henle and collecting tubules. These, again, are key structures involved in concentrating urine and transporting it to the renal pelvis. Examine the lumen of these tubules, where the filtrate flows, and note the surrounding tissue layers, including the adventitia, which provides structural support. In the bladder histology, identify the rugae, which are the folds that allow the bladder to expand as it fills with urine. This slide here represents the histological view of the ureter, and it highlights its layered structure. Begin by focusing on the lumen here. This is the central space through which urine flows. And then surrounding the lumen is the transitional epithelium that we saw earlier. And this has a basal layer that anchors the tissue and a superficial layer that accommodates stretching as urine passes through. Beneath the epithelium lies the lamina propria. The lamina propria is a connective tissue layer, followed by, then you will see the smooth muscle layers. So over here you have the lumen. This is the lamina propria. Then you have this smooth muscle layer. Notice the circular and longitudinal muscle layers, which generate peristaltic contractions that propel urine toward the bladder. And then outside those muscle layers is the adventitia, which contains supporting structures such as nerves, arteries, venules, and adipose tissue. In some sections, you can also see fibroelastic connective tissue, sometimes referred to as subserous fascia, and this layer provides additional structural support. So seeing how all of these layers are organized and how they work helps you to see microscopically how the ureters are both strong and flexible. And this helps with transporting urine while withstanding repeated stretching and contraction. Let's look at the bladder histology figure here on the left. So you would again start by observing the lumen of the urinary bladder, right? This is the space where the urine collects. The inner lining is the epithelium, specifically transitional epithelium, which allows the bladder to stretch as it fills. And then beneath the epithelium is the lamina propria. So these are layers we saw in the ureter. Again, this is the connective tissue layer that gives support. And then surrounding that layer is the smooth muscle layer. Again, very similar to what we saw in the ureter. Now in the kidney histology figure here on the right, notice the distinct regions of the cortex and the medulla. The cortex contains the renal corpuscles and the bulk of the nephron structures responsible for filtration. And then the medulla is organized into pyramids that house loops of Henle and collecting ducts. So this is where the urine is concentrated, right? This slide is a histological view of a renal corpuscle. There's a couple of them. This one's a bit more zoomed in than this one here. Again, the renal corpuscle is the initial filtering component of the nephron. At the center is the glomerulus, a tuft of capillaries. You can't really see it here. You can see the outside, the Bowman's capsule, but inside is the glomerulus, which again is a tuft of capillaries where blood plasma is filtered. And then surrounding that is the Bowman's capsule, which is that double-walled epithelial structure that collects the filtrate and directs it into the proximal tubule. Then within the glomerulus, you can also observe the mesengium. The mesengium is a network of specialized cells and supporting matrix. Mesangial cells provide structural support to the glomerular capillaries and also helps regulate blood flow within the glomerulus and contribute to clearance of any kind of debris that would be in the urine. In lab exercise 8.3, you'll complete an interactive web activity to learn about urinalysis and dipstick testing. Begin by using an internet-connected computer to go to this website here. Make sure you read the introductory information provided on the site to understand the principles of urinalysis, the types of substances that can be detected by dipstick testing, and the significance of each parameter. And then once you're familiar with the background, click the green button to launch the simulation. You'll then test 5 different patient samples by virtually dipping the test strip and then comparing the colors that you see on the test strip to the provided chart. Be sure to record your findings for each patient accurately, as this will help you to interpret urinalysis results and understand how these tests are used to assess urinary system function and detect potential abnormalities. And then, of course, this exercise is going to reinforce your understanding of urine composition and prepare you to apply these concepts in a clinical or laboratory setting in the future. So this is just a screenshot of some of the steps. You'll go to the chemical exam first, and it's going to take you through all of these different steps of the urinalysis. And then you can always go through and click some of these background informations if you need to review some of the terminology. That would be here. It's a pretty straightforward simulation. I think you will enjoy it. This is just an example of the color changes that you may see and what they mean. So you can compare your dipstick with these colors to see if you see any abnormalities. Okay, well, that's going to be it for the Lab 8 video. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.